Welcome, everyone, and thank you for joining us for today's session, Pioneering Our Understanding of the Human Brain. It is my pleasure to introduce to you Dr. Nader Pouratian. Dr. Pouratian is an Associate Professor of Neurosurgery and Radiation Oncology and an affiliated faculty in Bioengineering and Neuroscience at UCLA. As a neurosurgeon, neuroscientist, and bioengineer, he has the unique perspective and training to integrate these fields and take advantage of the unparalleled opportunities presented by neurosurgery to study human brain function and design novel neurotechnologies. For a complete biography of our speaker, please visit the Biography tab at the top of your screen. Dr. Pouratian's talk is entitled Early Feasibility Study of a Visual Cortical Prosthesis for the Blind, the Orion Visual Prosthesis System. My name is Dr. Roshan Akashamyun, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. We would like to extend our special thanks to the Brain Initiative program directors, and in particular, Dr. James Knapp, for their efforts in organizing this session. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting your questions during the presentation. Simply click on the Ask a Question box located at the far left of your screen and type your questions in the drop-down box. Our presenter will respond to your questions via email following the presentation. The presentation is educational and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. Now, without any further ado, please join me in welcoming Dr. Nader Pouratian.
Hello, I'm Nader Pouratian from UCLA Neurosurgery. Thank you for this opportunity to speak to you about our early feasibility study of a visual cortical prosthesis for the blind using the Orion visual prosthesis system. I'll start with some disclosures. I have been working with Second Sight on this project over the past 6 years. Have had some grant support from them, and this is now a collaborative project funded by the NIH. I also do some consulting work for other companies in this space. So I want to start with the need. Why do we do this type of work? 39 million people worldwide are legally blind, and much of that blindness is due to unpreventable causes. In terms of treating blindness, there is a lot of work in terms of accessibility, with iPhones and other devices to improve quality of life. But in terms of restoring vision, there's really only one device that is FDA approved. It happens to also have been created by Second Sight, which is the Argus system, which is a retinal prosthesis, which couples a glasses with video camera, a video processing unit, and wireless connection to an array that is implanted on the back of the retina. That is approved for retinitis pigmentosa. There's some trials for age-related macular degeneration, but really it's only for people whose retina has some function left. And so its generalizability is a little bit more limited than we'd like. So the goal here is to develop a cortical prosthesis for previously sighted patients in order to make sure their visual cortex is developed who have no or bare light perception. Of course, anytime you develop a new device, there are safety concerns first and foremost. You want to make sure you're helping people and not hurting them. So there's implant-related concerns. There's concerns about seizures with cortical stimulation. We know that cortical stimulation can induce seizures. Secondarily, once we get those safety concerns addressed, there's practical concerns. Is it surgically practical to do this? Is it a way that can be adopted widely? And then clinical utility. What can people see? What can we help them see? Do they have to see the way that we know vision to be? So I want to explore some of these concepts, putting it in a historical context as well. We're not the first group, nor will we probably be the last group who does this type of work. Brindley and Lewin did a lot of work in the past with chronic implants in some patients, being able to place an array over the visual cortex in a with a subdural array, mapping out the visual space. But unfortunately, a lot of their work was limited, and much of it wasn't published due to untimely death. And so there's still a lot of work to be done. Again, a lot of research is also done in sighted patients and people who have epilepsy who happen to have arrays over the visual cortex. This is just one example from a group at Stanford showing that there is a very good prediction of the size of a phosphene that can be elicited in a sighted subject based on the current density that might be delivered by an electrode at any given spot. I don't want to get too much into that because I want to get into the practicalities of what can happen in a blinded subject, though. So when we started this work, one of our initial preliminary studies was actually in a blind subject. We thought we needed some chronic experience in a patient who is blind. We took an off-the-shelf device, which is the NeuroPace responsive neurostimulation system, in a 30-year-old patient with a history of Koyanagi-Harada syndrome, in order to see what she could see and how we can do this chronically and how reliable this might be. We did systematic manipulations. And this says over 10 months, but we've gone beyond that 10-month period at this point. I want to share some of those results. In this subject, because this was off the shelf, it was not purpose-built for testing blindness or testing vision in a blind subject. We had 2 electrocardiographic strips, each with 4 contacts. We have 8 contacts. And here you can see that each stimulation at each contact was able to produce a visual phosphene. It's a different size, different shape, different location for every area that was stimulated. And it respected the horizontal meridian as we'd expect according to the Calcarean sulcus. So this was very encouraging with consistent localization over time. This is an example of the shape and location of percepts across sessions, showing that when we stimulate a certain area, across sessions, they would see it in the same, or this particular subject would see it in the same spot over months of testing. So that consistency over time, the lack of plasticity is actually a good thing for developing a visual cortical prosthesis. We also learned a lot about the stimulation biology of the visual cortex. And this is something that came out, there's a lot of literature about what it takes to activate neurons. And what we found was that the stimulation parameters for phosphene production shares 2 basic characteristics of the strength duration curve of neuronal excitation. And again, without getting into too much detail, I'll say that these slides show that there's a Rio-based current, or a minimum amplitude of infinite pulse width frequency that is present. So there's a sort of a minimum current that must occur in order to elicit a response or a phosphene. There's also, again, a minimum charge that's required to elicit a phosphate, and the extra charge increases linearly as the pulse width or frequency increases. And those are illustrated here. These are very important concepts in terms of determining how a chronic stimulation system would have to be programmed and adjusted. Perhaps the more important slide is getting some insight into how these phosphene perceptions are changed by different stimulation parameters. So although you might think that it's total current that affects how people perceive a phosphene, what we see here is that the charge per trial is not what determines the absolute intensity. Rather, you could see, for example, under A1, the dotted line shows the perceived intensity of a phosphene as the frequency is modulated, whereas the solid line shows the perceived intensity as the amplitude is modulated. You can see that for the same charge per trial at approximately 10 microcoulombs, the perceived intensity can be dramatically different. And we see that in almost every single contact. So it's not just charge that we're interested in. Amplitude modulation and frequency modulation can differentially affect patients' perceptions with chronic visual stimulation. So this work was really essential in providing us the motivation to really charge forward with a purpose-built visual prosthesis system, which is called Orion, which leverages the existing technology from Argus, the same idea of having an implanted array wirelessly connected through a RF coil with a video processing unit, glasses with a video camera. The advantage of a visual cortical prosthesis is that it bypasses the optic nerve, it goes directly to the visual cortex. We did some preclinical testing to prove the safety of this implant in terms of large animal studies to confirm that there wasn't any adverse effect from the implant and stimulation. And then we, of course, did the trial that I just showed you with a chronic stimulation with a non-purpose-built device. This is, we're fortunately funded by the Brain Initiative. This shows schematically what the implant looks like. And in the first panel, you see the glasses with an RF coil and a video processing unit in the corner. The two brain schematics show the roughly triangular shaped array over the medial occipital lobe with a cable and a skull mounted array where we couple to and communicate with the device. This was based on cadaveric, large animal, and feasibility studies. And at this point, using this device design, we've implanted 6 subjects across 2 sites. The plan at this point is to follow these 6 subjects across the 2 sites at UCLA and Baylor, with the neurosurgeon leading at Baylor being Dr. Dan Yosher, with a primary outcome of safety, but secondary outcome of really assessing the feasibility, the ability to produce phosphene, long-term functionality of the device, and benefit in terms of visual function, functional vision, and quality of life. This is an example of what the implant looks like. It's a U-shaped incision. There's a small craniotomy seen in the middle panel where the cable runs into the inner hemispheric fissure where the array is placed over the medial occipital lobe. You can see the device and the RF coil to the left, and that's the incision there. As with any technique, we start a little bit larger with our approach in order to make sure feasibility and anticipate that this will get smaller over time. To date, with the UCLA subjects, we have an average age of implant of 50, 1 female, 3 males, and the causes of blindness are shown there. Trauma, pediatric blindness, someone who had a pediatric condition that evolved into complete blindness, and glaucoma. And that highlights really how this device is potentially useful for multiple diagnoses rather than Argus, which is currently only approved for retinitis pigmentosa. Implant duration, the shortest duration at this point is 9 months, and we have someone who's implanted over a year. And we're very happy with the results so far. As I said, safety is the major issue. And there has been 1 serious adverse event, which is a seizure, which has been resolved. That's something that was anticipated could be a problem. It's been in a single subject. We've had 4 non-serious adverse events as well, with hand twitching and a visual aura, as well as 2 events of headaches. There's 2 patients who've had no adverse events and 3 subjects with no serious adverse events in the UCLA cohort. In terms of our thresholds, this is something that we need to understand so we know how much power a device like this needs to have. Thresholds are determined based on being able to see a phosphene 50% of the time. You can see the average threshold is right around 2 to 2.5 milliamps across subjects, and that has been consistent over time and across contacts. As you can see in subject 1, 56 out of the 60 contacts are able to elicit a phosphene, 60 out of 60 in the second subject, and 59 out of 59 and 59 out of 59 in the third and fourth subject, showing very high reliability of producing phosphines. So we've done some preliminary studies in light localization, staring at a screen with a bright box where patients have to point to the light, and early testing has been very positive. Almost all of our subjects are able to do that with high reliability, as well as direction of motion testing. More details will follow as we analyze that data and present it in subsequent presentations. Our research focus in the first 6 months has really been to establish the safety, establish what our thresholds are, and then create video settings and establish stimulation safety settings so that people can go home with these devices. We've cleared all of our subjects at UCLA for home use, so they take it home and use it up to 2 hours a day to explore their home and see how the device can be used effectively. We've also initiated rehabilitation sessions, so it's very important to realize that these patients aren't going to have normal vision restored, but they need to learn to adapt to this new form of vision. And it's not to say that it's not useful, but it's something new. In some ways, you can consider it like sensory substitution, and they need to learn how to use it and identify the pathways to making it the most effective therapy possible for them. One of the issues we've been focusing on is this issue of spatial mapping. In the Argus 2 with a retinal implant, there was a one-to-one spatial mapping, meaning if you stimulate in a pattern that looks like an H, you would anticipate that a patient would see in the form of an H. As we know, the visual cortex doesn't have that type of mapping. And so just because an array looks a certain way, stimulating a certain pattern or an H, as we see here in blue dots on the array, does not translate into an H in the visual space because of the organization of the visual cortex. This is an area that we are studying extensively. We're trying to understand how we should do the spatial mapping in order to make the device as useful as possible. Note that I already said that the light localization is very good. Direction of motion testing is already very good. So even without having a deep understanding of the optimal way of doing spatial mapping, we still have very promising results. But we need to develop an algorithm for efficient and accurate spatial mapping in order to make this useful. And one area we've looked at is from the imaging literature. We have a lot of information about how polar angles are mapped in the visual cortices, as well as eccentricities mapped in the visual cortices. And here you could see an Orion array with the white dots overlaid in that area. And one of the questions is, how reliably can we predict where the phosphene will be based on these population level maps? Are these population level maps the best way to map out the visual space and get the device up and going, or do we need individualized maps? That's part of the investigations that we're doing currently to better refine the system for home use. I wanted to start closing out my presentation with a couple examples of real world use and patient experiences that will help you really understand what this is like for patients. So I'll take some time to read these. These are feedback from the vision rehab specialist. So the first one, he was able to find the cue ball with no problems on the table. And note that these are patients who have no light perception or bare light perception, wouldn't have been able to do this before. He's able to tell the cue ball from the blue 10 and also from the balls with the stripe versus the cue ball. He could find the racked balls at the other end of the table too. Here's another example. We found that looking forward is where the Orion shines better for the user as they can detect upcoming objects. He's able to see cars parked on the side of the street, openings in the sidewalk, up into driveways, etc. Again, so the device is not the vision as we know it, but as a visual aid could be extremely useful, especially if integrated with other kinds of visual aids. The next one, when working throughout her apartment, working throughout her apartment building, she's able to tell where I was located and when standing in front of a 10-foot wide light wall without visual clutter. She's also able to correctly determine whether I was traveling from left to right or right to left along this well 7 out of 10 times. Again, pretty amazing considering these people have no light perception for the most part. Finally, he's able to order patterns from small checkers, big checkers, and white cloth. There's a ½ inch difference in checker size in the patterns. So I think these are very hard to quantify. In clinical trials, we're very focused on quantification, but the quantification that we do in the clinic setting with formal testing is not necessarily representative of real life, and we think these real life examples are really where there is significant value. And I think what we're finding is that subjects are finding success with Orion for everyday tasks. I will say that the amount of success is variable, and not every subject is able to use it the same way. It has to some extent to do with expectation management. So I'll start with what are our conclusions. Primary visual cortex stimulation is feasible. We've seen that with our first patient who didn't have a purpose-built device with the NeuroPace system, and we're also seeing it with early results from our feasibility study here. The simulation parameters are predictable based on basic science insights into the physiology of neuronal excitation. Phosphines are reproducible, they're consistent over time. We're now trying to better understand the modulation of these phosphines, temporal factors. So how quickly can you stimulate to develop a constant perception? Or if you want to develop a flickering perception, what are the temporal parameters? We need to understand frame of reference and then real life applications. Some other issues that I will just briefly talk about, and I've mentioned already, these temporal issues of flicker fusion experiments are in our future that we need to do. Spatial resolution issues, how Do we need to make the array even more precise, or is this basically what we're going to get? What further work do we need to do? These are questions we need to address. I've talked about spatial mapping. The importance of rehabilitation has become very clear over time because this is not a plug-and-play solution. People need to learn, just like with any rehabilitative device, how to use it optimally. And then this idea of sensory substitution. Finally, there are ethical issues with restoring some kind of sensation or ability to patients, as well as the people who enroll these trials and the expectations they have, and there's something we need to consider very carefully. So I hope I've been able to give you some insight into our work in the visual cortical prosthesis. Thank you for your time.
Thank you for that exciting talk on this cutting-edge technology, Dr. Pouratian. As a reminder, our speaker will follow up any questions about his presentation by email. Next, we have Dr. Michael Beauchamp and his colleague, Dr. John Magnotti, who will be giving another innovative presentation on RAVE, R analysis and visualization of intracranial encephalography. Hope to see you there.